So, what we're going to talk about for this video is membrane transport. You want to understand these mechanisms because they're going to come back when you go into certain processes and you want to know why something works the way it does. So, eventually you'll talk about the sodium potassium pump. But before we do that, we want to know that there's a few different types of transport. So, you got to know that the plasma membrane is selectively permeable. All right, it's selective. Not everything can go in and out. It's kind of like a bouncer at a club. You're gonna want to see and recognize something before you flag it in or out. You'll let some things come in and some things go out, such as nutrients, wastes, and ions and other things. But it's going to prevent other things from leaving the cell or entering, such as a protein or a phosphate. Because those are important, you want to keep them in. They have regulatory roles and other functions that you don't want to lose. So there's two ways to classify the moving of substances, active and passive. Passive, that does not require energy directly, whereas active mechanisms do require some form of energy currency. Passive mechanisms, those include filtration and diffusion, right? And a type of diffusion would be osmosis, right? So let's talk about filtration. Filtration is when you have a um, particle that will enter through the selective permeable membrane by a hydrostatic pressure, right? Which is the force that is exerted due to water. All right, so that would be filtration. If we talk about simple diffusion, that is the net movement of particles from a place of high concentration to a place of low concentration. So if you have 100 particles here and only 20 particles, the same particle, it's going to travel down to establish some type of normalcy. Um, you talk about simple diffusion, and you want to know that there's a concentration gradient at play here. That's what's causing. There's a difference in concentrations, so there's a gradient, which means you're going to see some movement. When you're talking about diffusion rates, there's a few things to keep in mind. The temperature, the molecular weight, the steepness of the gradient, the surface area of the membrane, and the permeability of the membrane. If you increase the temperature, you are going to increase the rate of diffusion. If something is bigger, heavier, you're going to slow the rate of diffusion. So that's why small ions and electrolytes, they pass so quickly. The steepness of the gradient. If it's 100 particles to 10, that's like a 90 difference. If it's 195, that's not that big of a difference, is it? No. The surface area. If you have a bigger wall that's permeable, more particles can come in and out. If that surface area is tiny, there's not that much room, there's not that much area for particles to move around. What about the permeability? That membrane and its permeability, those are going to be different for different membranes. You know, also, ions may have different permeabilities. Potassium will diffuse more easily than sodium, right? Nonpolar hydrophobic particles, they can pass through the plasma membrane because a good amount of that plasma membrane is also nonpolar hydrophobic. You know, those lipid portions. Another type of transport you want to talk about is osmosis, which is the diffusion of water down its concentration gradient. So, whereas simple diffusion is talking about other particles, osmosis is talking about water from high to low. So, how does water enter a cell? A lot of water, like most amount of water that will be coming and going, those will come through what you call an aquaporin. Aquaporin. Porin, like a pour, aqua, water. Cool stuff. So Those are specialized channels for the passage of water, like we said. So if you have more of these channels in the membrane, you're going to see an increased rate in osmosis. It's like opening up more highways just for one type of vehicle. That you're going to get a lot more vehicles going through if you have a lot more highways for that car. Let's see what else we have. Alright, so the direction of osmosis is from a more dilute solution to a more concentrated one. And when we say that, it's important to realize that water follows ions in polar objects. So if I have like 5 grains of salt and I have like 1,000 grains of salt, water's going to go towards there's more salt. It's going to follow those ions, and that's what it wants to do. That's the property. So when they say, you have a beaker with 10 and a beaker with 100 particles of salt, and they're connected by a selective permeable membrane, water is just going to go through and towards the area with high salt. So that's one thing to keep in mind. Carrier-mediated transport. Okay, so these carriers act like enzymes in some ways. Basically, they will move a particle and they will either help it go down its concentration gradient or above its concentration gradient. And these carriers do exhibit specificity, so they are particular. It's not going to just take anything. If it's a carrier for a particular protein, it's not going to just grab some fat and just pull it through. There is a transport maximum to keep at play, which is the maximum rate, how fast something is going in and out of the membrane. Let's see what else we have. Carrier-mediated transport will also include a symport, which is carrying 2 or more solutes through a membrane, a uniport, which only carries 1 solute at a time, an antiport, which carries 2 particles but in opposite directions, one in and one out of the cell. You have facilitated diffusion, which is the carrier-mediated transport through a membrane from high to low, and you have active transport, which is from low to high, because it's using energy to go against the concentration gradient, so against the steepness of the curve. What's an example of active transport? The sodium-potassium pump. It's one of the most talked about ones in physiology. I know we talk about it a lot. So, vascular transport. So, vascular transport is moving large particles, droplets of fluid, waste, whatever, through vesicles, which are basically the same constitutions of the membrane. And you can do endocytosis, endo meaning come inside, or exocytosis, exit the cell. Phagocytosis is a form of endocytosis, which is when you eat something, like a large particle, whereas penocytosis is taking in a small particle, so you're drinking, because you eat big things, but you drink little things, right? So that's how you differentiate between phago and penocytosis. So that seems to be the gist of membrane transport. You just want to note that you have active transport and you have passive transport. And then within passive transport, you may have facilitated. You just may have, simple diffusion. And then active transport, you have things that are going against your concentration gradient. So that would be that.